So what's happening is these two worms, the bacteria in these two worms, are taking the hydrogen sulfide. Not only are they making sulfur out of it, but that energy, they're actually using it to not only to make ATP, but to also synthesize a number of other biologically useful compounds. The carbon dioxide is being fixed, and it's being fixed in a way that sugars can be made, and other compounds and other carbon compounds can be synthesized by these organisms that really don't have any other means of obtaining energy except through the oxidation of sulfur. So that makes these things very, very unique. And the tube worms are somewhat different than some of the other organisms. There is a clam called Calyptogena, Calyptogena, another one of the vent, hydrothermal vent community members. And for instance, for that particular organism, sulfite is the only effective ATP. It's the only effective sulfur species for stimulating ATP production. But with the tube worms, we find that both sulfite and sulfide, like hydrogen sulfide, actually stimulate ATP synthesis. So there might be different organisms that are involved in these things. And in the Calyptogena, in this particular clam, the bacteria, rather than being in a trophosome, are actually located in the gills of the clam. So it doesn't have this special organ that the tube worm has. The rate of carbon dioxide fixation is approximately 13.5 nanomoles, that's billionths of a mole, per milligram of protein per hour. And if you add things like thiosulfate, you can actually triple the rate. You can get the rate up to about 40 nanomoles per milligram per hour. So it's possible by using some of these other sulfur species to actually increase the amount of carbon fixation and also the amount of ATP synthesis that's taking place. There have been some studies that were done looking at enzymes that are actually isolated from the trophosome, and some of the enzymes that are involved are an enzyme called APS reductase. APS is adenosine phosphosulfate. It's like adenosine monophosphate, AMP, with a sulfate, with an SO4 attached to that single phosphate group. There's another enzyme called ATP sulfurylase, and this is an enzyme that actually forms the APS, the adenosine 5'-phosphosulfate, by taking AMP, adenosine monophosphate, and adding sulfate to it. So that's how it's formed. And the APS reductase, what that does is it actually takes the sulfate and reduces the sulfate to sulfite, from SO4 to SO3. There's another enzyme called rhodanese, RHODANESE. And I was surprised to find this enzyme because the enzyme rhodanese, one of the things that it does is it actually adds elemental sulfur to things like cyanide. And this is where I was telling you before, one of the ways that you can detoxify cyanide is you can take cyanide and add sulfur to it, and you can make something called thiocyanate, CNS. And this species tends to be pretty non-toxic. The reason hydrogen cyanide kills us is this requires this enzyme called rhodanese. Now, the problem is we've got plenty of rhodanese in our cells, and when cyanide is introduced, we've got the rhodanese to do the job. What we don't have is a free source of sulfur. So when you want to treat somebody for cyanide poisoning, in addition to putting in something that will oxidize the hemoglobin up to methemoglobin, and they usually use things like sodium nitrite, which will do the job, or sometimes they'll have an individual sniff a capsule that contains a volatile liquid called amyl nitrite. But the other thing you can do is you can actually administer sulfur rather, and it's done either by using cysteine, which is that amino acid, or another real fast way of getting it in is to use this species, sodium thiosulfate, and that would actually be injected directly into someone's veins, and by doing that, it's possible to actually save somebody who's been poisoned by cyanide. So you have rhodinase and APS reductase and ATP sulfurylase in the trophosomes, in the bacteria. And then surprisingly, you also have, or maybe not so surprisingly, you have two other enzymes. One of them is called ribulose 5-phosphate kinase, and this actually forms from the sugar ribulose. It actually phosphorylates the ribulose, puts a phosphate group on it. And then there's another enzyme called ribulose 1,5-bisphosphate carboxylase oxygenase, which has the shortened form. It's called Rubisco. Rubisco, which stands for ribulose bisphosphate carboxylase oxygenase. So Rubisco is an enzyme that's actually involved. Those of you that have had a little biology and have looked at photosynthesis know that there are two cycles in photosynthesis, one light cycle and another set of reactions that used to be called the dark reactions, although they're no longer referred to as dark reactions because they take place either in the light or the dark. But the light reactions basically split water into protons and oxygen, and what the other reactions do, which are part of something called the Calvin cycle or the Calvin-Benson cycle, what they do is they actually fix carbon dioxide. So all the carbon dioxide that's in the air, when it's taken up by plants, they use this enzyme Rubisco to actually incorporate the carbon dioxide and make some of these sugars that we talked about. So it's an extraordinarily important enzyme. And there it is in with all these other sulfur-fixing enzymes, which has led scientists to believe that all of these reactions are actually coupled. So the energy that's derived from the sulfur reactions is actually being utilized to drive the Calvin-Benson cycle, where normally the energy would actually come from electron transport that was derived from some of these photosynthetic reactions. So these are very, very important chemical reactions that are taking place.